The synthesis of a polypeptide begins after a messenger RNA binds to a free ribosome in the cytosol. The polypeptide contains a signal sequence that is recognized by a signal recognition particle, SRP. The SRP, after finding a lonely ribosome, binds to the signal sequence in ribosome. Following this, the SRP and the ribosome then bind with an SRP receptor found along the exterior of the ER membrane. After binding, the SRP receptor moves across the membrane to the closest protein translocation channel to cross through the ER membrane into the lumen. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. After passing the ribosome to a translocation channel, the SRP complex is released to repeat the process elsewhere. While the protein chain is threaded through the membrane as a large loop, the signal peptide remains bound to the channel. Translocation continues with the growing polypeptide continuing into the rough ER. Oligosaccharyl transferase transfers an oligosaccharide from a doylecyl phosphate to an asparagine on the growing peptide. Once it is fully translated, the signal sequence is cleaved off by a signal peptidase on the luminal side of the ER. This causes the signal peptide to be released from the translocation channel into the membrane and then rapidly degraded. The plug then rebinds to the ER lumen, closing the inactive channel. Now protein folding continues. Glucose molecules are cleaved off the end of the oligosaccharide, and in final phase, only one glucose molecule is left on the oligosaccharide. This allows cholinesterase and ER chaperone to bind to ensure complete and proper folding along with protein disulfide isomerase, which helps form disulfide bonds in polypeptide. If it is folded correctly, final glucose molecule is cleaved off. Transport vesicles pinch off of ER and move folded proteins from rough ER to the cis Golgi. The Golgi complex is split up into three parts: the cis, medial, and trans Golgi network, and between the two cis, medial, and trans cisterna. The cis Golgi sorts protein so it can continue to the next Golgi section or to be sent back to the ER for protein synthesis as it goes from the cisterna portion of the Golgi complex. There are distinctive sets of enzymes at each cisterna. If the protein is correctly folded, the sequence of sugar-adding enzyme in the Golgi complex determines the sequence of sugar addition to glycoprotein traveling through the Golgi. Vesicles move towards the target along the cytoskeleton with the help of motor protein and receptors. Then the vesicle is tethered by the attachment of tethering proteins to the vesicle rab, fastening it to the membrane rab protein and drawing it in. Once the tether protein grab onto the vesicle docks at the target with the help of the V and T snares, and in the fusion step, the membranes are fused together with the force of the intertwining of the V and T snares. As the vesicle and membrane are fused together, the receptor can release the cargo protein for execretion.